Okay, welcome back everybody. We'll make a start on our next session. So having had the presentation before the break, partly from me, you're now stuck with me as chair for this session, but there won't be very much from me. We've got three really great speakers, really looking forward to hear what these three people have got to say around this kind of idea of how we build the bioeconomy. So to start us off, to talk about how we can kind of enable innovation in engineering biology, it's Thierry, Goff, Commercial Director of the National Measurement Laboratory. Thierry.
Thanks, James. Thanks to IBIC to have me today. I'm going to talk about metrology, which is not a topic that is talked about a lot in that kind of conferences, but very important, and also a great believer of networks, because to be able to exploit the potential of engineering biology or biotechnology, innovators will need to talk to people they are not used to talk to normally. That can be the regulators, that can be standards, that can be investors, etc. And we need to be able to connect that wider regulatory system with innovation to be able to push and exploit the potential of engineering biology or biotechnology in the UK. It's quite clear that the UK has got an amazing research base, and there is a drive now from government, DCIT, so the Department for Science, Innovation of Technology, or UKRI, to have a bit more emphasis on exploiting the potential of that fundamental research and create commercial product to drive economic growth. And it's also good for the planet. So thanks for having me. So I'm going to raise a little bit of awareness of metrology and standardization. And as a quick poll, I'll turn around and say, how many of you have heard of metrology before? It's not too bad. So I'm going to raise awareness of it and how we actually support innovation rather than being a barrier. And I'll talk about standardization, because standardization is probably even worse. If I ask how many of you are aware of standards or how it works in the UK? CPI, yeah. So I'll explain a little bit then, and I'll generally start with this to explain what we mean by metrology and standardization, because there's no real point for me to carry on if we don't understand what we're talking about. And you probably agree that metrology, which is the science of measurement, is quite key in engineering or manufacturing. It's quite easy to understand that if you are talking about the car industry or any industry, they will need very precise measurement and standardized parts to come together to get a consistent product, and they need methods to control the process to manufacture their product, good control of the raw material, etc. So it's actually key in engineering. However, some work to do in transformative technology like synthetic biology or engineering biology. The metrology will provide is this idea of providing a framework for traceability. One example of that for weighing in the kilogram is in the past, in Paris, in the BIPM, we were keeping the kilogram, and then all of the countries in the world were calibrating their kilogram against that main standard. So we could have comparability, so you had no difference in time or location, and enable reliability of data. And you agree with me that probably in innovation, you need reliable data to be able, or reproducibility to be able to push it down the innovation pipeline to commercialization. Standards is a complete misconception of what standards are. So the second bullet point is approved by a recognized body. You've got ISO at international level, you've got CEN at European level, and you've got probably, BSI at the British level. But they are the recognized body to approve it. Those standards are developed by experts like you. And actually, the standards can start with an SOP as a network or a group of people that are expert in their measurement. And that standard will provide guidelines for not only the final product, but also the process to define the characteristic of the product. So it can be repeated, transfer, better consistency. And what we focus in the National Measurement Lab is both the metrology and support the standardization And the two together is this idea of improving reproducibility in innovation and more DPTL efficiency. So better control of the raw material, better methods to control your process, and methodology at the end to check the specification of the product, so you don't have variability between one batch and another, which is quite important for commercial product. So it is really critical for producing a reliable and safe product, but it's not always considered at early stage of innovation, TRL 3 and 4. So that's what we have been involved in the past few years, is to raise awareness of metrology amongst innovators to ensure that it's at least considered, so the translation is a little bit more efficient. So who are we? We are part of the national measurement system. It's a bit different from other countries. Our designation of the institute is chemical and biomeasurement, so we are all about the measurement in that space. But the national measurement system also involved a national physical lab for physical measurement, NIPSC for biological standards, or NIPSC or MHRA is a new name, and others like NEL for flow. We are like a university assessed externally by international experts, and we are internationally leading or internationally competitive in the space that we are covering. We are located in Guildford. We've just moved there. We've got about 150 scientists split into 4 main labs covering organic analysis, small molecule and peptide, protein, etc. Inorganic, covering elements, species, nanoparticle, and the molecular biology lab supported with platforms such as digital PCR and qPCR looking at DNA or nucleic acid quantification. We work across sector, which is quite helpful for today, so health, diagnostic, advanced therapeutics, environment, sustainability, and food. We are producing or developing and validating methods for all sorts of applications. We validate them, we produce reference material for innovators to check their methodology, training, consultancy, and we also advise the government and provide advice to the industry and academia on their measurement need. We are, since September, eligible to receive UKRI funding, so we can collaborate with universities and embed metrology in their research, and we have been doing that in the past 6 months quite a lot. We have got other unique roles, but we're not going to delve too much into that right now. What you're probably interested is to find out what kind of analytical platform we've got and how to access it. So we are an R&D lab. We don't do routine testing. And we are not expert in an application. Our measurement scientists, the 100 measurement scientists that work on site, are platform experts, and they're able to apply their expertise across multiple applications. So we really welcome the challenges of difficult things, like trace level, difficult matrices, and small sample size, new analyte, screening of impurities, etc. That's what we like to do, and we've got a platform split across the molecular biology and the chemistry space. In terms of the biology, we're using mainly qPCR and digital PCR for the nucleic acid, so the quantification of DNA or RNA, sequencing, immunoassay, flow cytometry for cell studies, and since recently we've got a benchtop nucleic acid oligo synthesis facility that we are setting up and validating, and we are hoping to use that to produce controls for the future. On the chemistry side, we are well kitted as well. We've got ICP for inorganic application. We've got a wide range of separation technique. We've got probably over 20 different mass spectrometers. We've got collaboration with Agilent, Waters, SciEx, Bruker, so all the manufacturer provider. We have got a QNMR for the purity of organic molecule and then the top ratio, and we can measure anything from element to trace metals, small organic molecules, nanoparticles, etc. So as I said, we are funded by the Department of Science, Innovation and Technology. And every 3 years we formulate a research program. That research program was aligned with the UK strategy. So engineering biology is obviously quite important. And we also align our research project with the need of our stakeholders. So that's why that network is quite important. Hopefully I will be able to have a lot of conversation and understand the measurement needs out there to potentially have some collaborative project or embed that in our research program to support you. So we are leveraging that expertise and that infrastructure, and we are working with our partner to design that research program. And if you look at some of the bullet point here, is going to be very similar to what is on the IBIC poster there, where I spent probably the last 2 years to go to that kind of conference and talk to innovators. And the first thing that strike me is probably the skills and the knowledge of metrology and personalization is not there, it is misconception. So we are spending quite a bit of time to understand what the skills require and develop some training program. And then we are also developing tools or methods or reference material, guides to support innovators to do good measurement in their lab. And what you're going to be interested in is how to access our infrastructure. Access to infrastructure is quite critical, not only measurement infrastructure, but also scaling up that we'll be talking about later on. Regulation, that's quite important, and probably James and Joanna will be able to confirm that it's probably quite a bit of time or effort to understand how to comply with regulation. So we are helping in the dialogue between innovators and regulators to understand actually what kind of measurement do I need to do to comply with those regulations. So in terms of skills and standard development, this adventure started about 3 years ago, where with the BBSRC we developed that training programs in metrology that is available for free if you talk to me afterwards. We did it in collaboration with Edinburgh, Norwich, Imperial College, UCL and Bristol. So quite a nice distribution across where we survey what's the new about metrology and standardization and develop that training program which is now available. It's a phased approach where we've got the basic explained in the one-day course, and then we've got some more advanced workshop when we understand what the innovators are doing at university, and I'm able to connect them with the measurement expert within the NML to make sure that they don't waste their time when they develop and validate their methodology, or if they don't have the kit, to access the kit within our institute. We are also collaborating, because we are a bit more known now, the CTT, we are providing the training for the one college by Oxford and Cambridge in engineering biology, and we are part of a number of proposals on the FOCO PhD program on engineering biology and AI. We went from not being very known to be part of many. So it's quite nice that the message is going through. In terms of standards, we survey our first cohort as well to know what kind of standards they would like, and it was quite clear that they wanted something about gene expression and quantitative measurement. So we proposed that standard to our stakeholder, and actually when we went to ISO, they thought that it was very interesting, and that standard proposed by 5 universities in the UK and other stakeholders is now UK-led. It's an ISO official item within the pipeline, and hopefully that standard will be official in a few years. The next step of our work in terms of training is we've just completed the training of the engineering mission hubs in the beginning of the year. So we did the one for plastic, P3B, IBIC, the environmental one led by Cranfield, and Elemental, the one in Kent. So we put them together And that was a good opportunity for them to exchange as well, and had some conversation with company about enzyme. And it was quite clear that they have got different needs, but they have been talking to each other, which is quite nice. And the next step is to try the one on microbial food and the Benno Center for Alternative Protein at Imperial College. And in Denver, talking to Susan Rosser at the Biofoundry to provide that training to her. We very quickly, within the NML, we're probably part of all of the standards that are being drafted at ISO level in biotechnology. We are the convener for the UK for the ISO TC276 on biotechnology. And because this is quite new, you don't have so many standards in biotechnology. We've got 35, which is not a lot for a potential multi-trillion bioeconomy. So it's something to do there. When you start into a field, you start with vocabulary and methods, and that's what we are doing: nucleic acid synthesis, gene delivery, lipid nanoparticle, etc., etc. We were also part of this group led by NPL and Imperial on Standard and Metrics for Engineering Biology. If you haven't read it, that's a good source of information about industrial standard rather than the ISO one. And we are also collaborating with a company, GitLife Biotech, to produce a new standard on barcoding, which is quite important in engineering biology because it's linked to biosecurity and make sure that all those engineered systems can be traced when they are deployed in the environment. Very quickly, when we put metrology in practice, we are split into three different themes: the therapeutics and diagnostic, the environment, and more the fundamental research, where we develop methods and also control material to support innovators to ensure that they have got a good system to control the experiment and also to determine the final product. So we are collaborating with the University of Kent on viral vector, applying mass spectrometry and digital PCR to ensure that the dose of those new therapeutics is known because of the implication risk if we deliver too much to the patient or not enough. We are collaborating internationally with our counterpart in the US on flow cytometry. We've collaborated with CPI and the Syngene therapy on scaling up, and on the DNA synthesizer, we're producing control for UPHSA for ensuring that the assay are still performing when you have got variants of concern for seasonal flu, for example. In terms of the environment, we've just started with Bristol and the Cyber Award under the EKRI BSRC on methodology to trace engineered system in their wind channel. So the idea is to use cyanobacteria to remove pollutants or other things, or application. And we focus on developing the methodology to make sure that when they are deployed, we'll go away to trace them in the environment. Very quickly, a few case studies where if you are interested in digital PCR and NGS, basically talk to us. In terms of molecular biology, we are expert in that field, internationally leading. And if you are using CRISPR, we can even exchange SOP to ensure that if you are using those platforms, you can make sure that the experiment or the preferred experiment is yielding what you want. A few things on regulatory support, and mainly around food. We are supporting a number of organizations from the DCID Regulatory Horizon Council. We are part of the sandbox on cultivated meat. We are part of many regulatory science network, and what a lot of people don't know at LGC, we are also doing the risk assessment for GMO on behalf of the FSA. We advise the government on precision breeding and what kind of technology can be applied to see the difference between natural and precision breed organism, which can be quite tiny. And we collaborate with a lot of institutions in the UK. So you're probably asking, okay, this is all great. How can we actually access your infrastructure? Because I don't have a mass spec, or I don't know how to use it very well for that application. In terms of industry and university, there are two main ways. If your research aligns with our goals, and engineering biology is one, we can maybe integrate it in our program. And if you are used to do grant, UKRI, or NIHR, or all sorts of other grant, we can draft the proposal. I spend lots of time writing grant application with university to get that money and embed methodology or method development in their research project. So we can provide something around knowledge exchange, transfer, method validation, or control materials that are not commercially available using the capability which are there. And just a brief acknowledgement of, and I probably don't have everything in there, so I apologize. Because that's the partner which we worked in the past 12 months. Thank you very much.
Brilliant. Thank you very much, Thierry. I'll invite kind of one burning question that someone may have. So I think for me, when you were looking at the case studies, how do you think that you can feed into kind of getting the regulatory networks and the regulators to understand what we're doing when we're bringing these innovations through? So it's a very vague question, but you alluded to it in your talk, and one of the things that you see is a lack of mutual understanding that perhaps gets in the way, and I think measurement might help.
That's a little side project that I've got, which is, you've got many networks around biotechnology or engineering biology, network in food, network in the environment, all sorts of network. I think what we are missing maybe, and that started with the sandbox exercise, where they put the industry in contact with the regulator to understand what kind of document or analysis we need to do. But what is missing more generally, and Rio is doing a good job, has just started that, the Regulatory Innovation Office, which is, they just put a contact name of and email addresses of people you could talk to within the regulator. But more generally, what's missing is a connection between the innovators, and I'm not talking industry generally, but also universities through the spin-out, and the wider regulatory system, which includes the standards, the regulators, and ourselves, the metrology. And I think probably what we can help in the metrology, because we're involved in the measurement but also the standards, is to maybe facilitate the dialogue between the innovators and the regulators of the standard, because we will have the scientific understanding.
Yeah, and I was kind of thinking, how do you get that kind of engagement? No, brilliant. Thank you very much.
Thank you very much.
So next speaker in this session is Jack Rowbotham, who's a RSC University Research Fellow in the MIB. He's going to talk to us about inorganic biocatalysis. Jack.
Thank you. Thanks very much, James. No problem. Thanks to IBIC as well for the slot. So I'm a recently established early career PI in the Institute of Biotechnology in Manchester, and my group looks at enzymes, particularly metalloenzymes, really interested in natural metalloenzymes and also hybrid metal enzyme catalysis. And ultimately what we're trying to do is do chemistry that's useful for society in a more sustainable way. So in my slot, I'm just going to give you some quite high-level examples of really past, present and future projects that I've been involved with that we've been trying to push towards an impact beyond the lab. So just before we do that, for those of you that I'm aware it's a broad room, biocatalysis, this is how AI renders biocatalysis, which I really like. Essentially, we're using enzymes as chemists to try and do very difficult chemistry. Often it's selectivity that we're after. And the guiding principles that we use are those of what we call the 12 principles of green chemistry, which try and keep a chemical process in some sense sustainable as possible. And actually, we find that when we look at biocatalytic processes, those that we're using enzymes, we're often ticking a lot of these boxes. But we have to be careful of assuming that just because we're using biocatalysis, we are in fact doing green chemistry. And sometimes things like atom efficiency is not always perfect in a biocatalytic reaction. Sometimes there's actually a lot of carbon-based waste that we might want to avoid. One of the ways around that is by using enzymes, especially enzymes which shuttle electrons around. And when we're moving electrons around, we don't have to be using carbon-based molecules to move electrons, so it's generally much cleaner. So I know better than to try and explain enzyme electrochemistry in a quick slot on a Tuesday morning. So I won't, but just to highlight a project that I was involved with as a postdoc before I came up to Manchester, where we're using two metalloenzymes that can pump electrons in and out to drive catalysis. By co-immobilizing these enzymes that can shuttle electrons around, we could drive some very selective chemistry, particularly useful for sort of pharmaceutical and fine chemical manufacturing, very cleanly, just using hydrogen gas. And so this, as a postdoc, I was involved in the spin-out of a company called Hydrogen, which some of you might be familiar with, spun out of Oxford under Kylie Vincent and Holly Reeve. And that really taught me a lot about what it takes to go from lab to spin out and from then onwards. And actually, a little case study within that, hydrogen has always been known for that asymmetric, very selective chemistry. But actually, one of the first projects I did, and I put a clipping of my lab book there, you can see it's September 2016, was a hydrogenation, so a nitro reduction to an amine. This is very sort of Victorian chemistry. It can be quite dirty chemistry. And actually, this process that we developed to do this chemistry very cleanly using hydrogenase enzymes, we only published it last year, I think it was August last year in Nature Communications. And what's nice is it's come with me to some extent up to the northwest, and now James Robinson, who I think are in the room today, are now trialling this enzymatic process for some of their chemical manufacturing. And so it was really nice to see that journey from what was essentially a 200 microliter reaction into now, and I hope James Robinson don't mind me saying, to their grubby hydrogenation reactors. It was a really interesting thing to be a part of, but I really learned that these things can take quite a long time. So with that, thinking about some of the newer chemistry that I'm doing now in my newly established lab, one area we're very interested in is CH activation, particularly inspired by a company called Oxford Biotrans that use these enzymes called P450 enzymes that can do really incredible selective chemistry using oxygen gas as the driving force. And in this case, Oxford Biotrans take this orange flavouring valenceine and transform it through a P450 enzyme into newt ketone, which has a grapefruit flavouring, with a considerable increase in the value of that compound. And so, again, this was a company that was spinning out whilst I was down in Oxford, and I kind of saw that selecting the product that you're making really can underpin the entire business. And I'll draw on that lesson later on in the talk. But I've been really interested in P450s for lots of reasons. They don't just do this sort of chemistry. There's a whole range of chemistry that they can do, have lots of really useful applications, but are underused in industrial biotechnology because they're quite fussy, they're notoriously quite unstable, they have generally quite poor turnover numbers. And so I think that improving that output, the sort of the lifetime of the enzyme is really critical to taking some of that very elegant chemistry that they can do and seeing it realized on an industrial scale. So that's what we've been doing in my lab. We've been looking at what the chemistry that P450s do. And actually, one of the things that's sort of glossed over a lot of the time is that these enzymes, just as those I showed you before with hydrogen, they require an electron demand, and it's a question that often academics tend to gloss over, is where these electrons come from. And actually, it's pretty critical to deriving the kind of output that we need from these enzymes to get them to the point where they could be commercializable. So I won't go through all the nitty-gritty of what we've been looking at, but we've been experimenting with different ways of pumping electrons into the active site of this enzyme, and trying to avoid toxic byproducts, so hydrogen peroxide, which is really the thing that limits the output of the P450s in a lot of cases. And through doing this, we've found strategies that are actually quite universally applicable for improving the turnover, the total turnover of this enzyme, and predominantly by improving the lifetime of the enzyme. And so this is work that we're looking to publish at the minute. But thinking about how we can use this in an applicable scenario, we've now got a project with Unilever, a prosperity partnership, which is a really big project involving a number of university partners, including here in Liverpool. And one of the work packages, we're looking at P450s for modifying polysaccharides, which is a sustainable way of making the sort of surfactant materials that Unilever require in a lot of their cleaning products. So this is a project that's really just getting off the ground at the minute, but it requires that improvement in the ability of the P450s before we can even start to think about using them on the scale and the ambition that Unilever have, which is really exciting. Then, if you'll forgive me for the slight non sequitur here, we're going to hop into the world of isotope chemistry. Now, isotopes are an interesting area of chemistry when you're working in biocatalysis, especially when you're thinking about commercializing something, because the demand for isotopically labeled products is often very, very small scale but high value, and very specific complex molecules, which is what biocatalysis does exceedingly well, especially when you're at the sort of university lab scale. So you can very quickly get to a product that has commercial potential. So I put this slide up here. For those of you that don't know, aren't familiar with isotopes, this is a nice example. So that glass of water there, you'll see the ice is sitting on the bottom of the glass rather than floating. And that's because the ice is made with D2O, deuterium oxide, rather than what normal water is, H2O. So that's a really nice illustrative example of what changing your isotope, because the chemistry is the same, but the isotope has changed, how that can lead to quite big changes to how a molecule behaves overall. And so with that sort of metaphor, we're looking at adding deuterium to molecules where normally there would be a hydrogen atom. And this has got really expanding range of applications. So that's an example of deuterium being the heavy isotope of hydrogen. And again, I know better than to try and explain the deuterium kinetic isotope effect in a 20-second window, but needless to say, it's a well-known property that swapping hydrogen for deuterium can lead to a longer lifetime of a molecule. So it's one strategy for stabilizing a molecule. And so that's now getting increasing awareness and applications in the world of pharmaceutical chemistry, whereby putting a deuterium onto a molecule can lead to a longer lifetime for that molecule in vivo, and therefore potentially lower dose rates for drugs. And so pharmaceutical companies are really interested in this approach of selectively adding deuterium to molecules. And that's something that we, as enzyme chemists, can see real opportunity for helping pharmaceutical companies to do this. And so I've got a project now with AstraZeneca where they have some targets that they're looking to try and stabilize, and so we're helping them to synthesize those using some enzymes. But getting into this world of isotope labeling, really we were just helping out a colleague in a protein NMR chemist who asked us, can you make a very specifically isotopically labeled compound for us because it's very expensive? I said, yes, that's no problem, we'll go away. We did it. And they were able to put it into a protein and get some structural data out of it that's relevant to kind of health and disease sciences. So we were just doing a job for them, just kind of making this molecule. But it was through that collaboration where we got talking about some of the demands for isotopically labeled compounds within life sciences research. And typical collaboration, we made this compound, it took us a few months. We took it to the collaborator, they said, thanks very much, and immediately said, can you make us something else? Which is how these collaborations tend to go. And we said, no, it's nothing to do with what we're doing. But actually, when we put pen to paper, we realized that there were some strategies that we could use to get to a different target, which was deuterated sugars. Which turned out to be the main cost to this particular researcher for their research. So they used heavy water, which is very expensive, but they tended to reuse that, so that it wasn't a major cost to them overall. Then there was the compound we made for them originally, which we made and they used, but again, it wasn't breaking the bank for them. It was these deuterated sugars that were limiting their ability to do research. And so for glucose, the most common sugar is about 300 pounds a gram. For the deuterated version, they were using about 4 or 5 grams every time they were doing an experiment. So you can see how those costs were rapidly adding up. So now through that, we've been working in the lab, and now we have quite a nice method of making deuterated sugars. So this is a spin-out venture that we're now getting off the ground called Deuterose. Glucose is the main product. We can make other sugars as well. And their immediate application is in life science research. But where we see real growth and opportunity is that there's an emerging field of deuterium metabolic imaging, so particularly MRI, where you can follow the deuterium in a normal MRI machine. There's no retrofitting that's needed. And you can look at where that goes in the body, how it breaks down in real time, in vivo. So it's very exciting. And actually, there's one of the places that's pioneering this DMI is the imaging units in Manchester. So it's really a great opportunity for us to link up. So in the interest of talking about scale up, we've gone from 10 mgs. We're at 10 grams at the minute. We've got customers who are interested when we get to 100 grams. So have me back in the next IBIC conference or the one in a couple of years' time. I'll tell you how we're doing and if it's worked or not. But thank you very much. Lots of people involved, lots of funders involved, and thanks especially to IBIC. And thanks to you for listening.
Awesome. Thanks very much, Jack. Again, I'll invite one burning question, and I invite someone to ask it so I don't have to try and ask a question about enzymes, because no one wants to hear that. Yeah, thank you.
So there's a lot of fields on enzyme engineering, which give including into the expression problems for activity.
I mean, I think that's one of the nice things of being in the Institute of Biotechnology is that there are people who are real experts in that, and I guess it depends which of the projects we're working on. So we do have some challenges with expression for the project that we're trying to scale up at the moment. And I think our strategy is just to try and talk to the people that are experts in it. Oh, brilliant. Okay, there you go then. Yeah, great.
Perfect. Thank you very much, Jack. Really interesting. Okay, so for our final speaker this morning, it's now the afternoon, before lunch would be a better way of putting it. Reihaneh Bashiri from CPI, who's going to talk about something that I was actually really looking forward to hearing about, how we can use kind of AI in IB, if you will, and really around how we can kind of look at getting more insight into fermentation processes. So Reihaneh, there you are. I was looking for you. Over to you.
Hi everyone, I'm Reihaneh. I'm Technology and Innovation Officer at CPI, Center for Process Innovation. And today I want to talk about a case study where we use fermentation data and federated learning to build a predictive model. When we talk about the industrial biotechnology, usually the scale-up and biomanufacturing are at the heart of the conversations. But when it is also important to consider that how we can make the biomanufacturing smarter. And it's important to understand during the fermentation process what happens and if we can anticipate some of the process events. And this is important because we can make some real-time decision and also it helps us to reduce the cost of offline sampling and plan for our resource management better, for example, if we have to consider night shifts and these sort of things. And also being able to predict what happens in the process helps us to de-risk the scale-up process. But we are not quite there yet, and there are different reasons for that. One of the challenge is that most of the data collection step is still manual, and most of the processes are relying on the offline data collections. The other problem is regarding the data privacy. Most of the companies or startups that they are developing a process, they are concerned about the IP, they don't share their data publicly, so that limits building these predictive models. The other problem is also related to the process itself. If you build a model and train it on the smaller scale data, it's harder to generalize that model to the larger scale, and that's one of the limits. The other problem is that there is still a lack of trust for the predictive models and also AI. So this requires the experts in the field of bioprocess and also data scientists to collaborate closely to see how they can improve this. For going forward, then we need methods that it can help us to develop these predictive models, and also it's scalable and keeps the privacy of the data for us. And this is where federated learning comes in. Federated learning, unlike the centralized model, enable us to train a model locally at different sites. So you don't need to share your data. You only train your model at your site locally, and then you share the updates of those models. And all of these models, they get aggregated and form a global model. And this global model later can go back to each of those devices, and everyone can benefit from this shared knowledge. So in the case study that we worked on in CPI, and also I apologize, I cannot really share the data because of the confidentiality, but it was coming from a fed batch fermentation, microbial fermentation process, and we wanted to see if we can predict two process events. Because the process was fed batch, it was important to see when the feeding should start, because at the moment you have to take offline samples. You have to take samples at different intervals and then do some lab analysis offline, which is really labor intensive and also costly. And the other was to predict the end of the fermentation. We used 15 process parameters, which they were coming from the online sensor measurements and also the offline samples at different intervals and lab analysis data. The data we used, they came from 3 different scales, from 150 liter, 750 liter, and 10,000 liter fermentation. We used Octai pipe as a platform to test the federated learning, and we used XGBoost model. Also, because we wanted to evaluate how federated models perform compared to other models, we tested individual models, which these models were only trained on the individual data set from each of the scales. And the central model was when we combine all of those different scales data, and then we train our model based on that, those combined data. And we had also fine-tuned federated, which it was like the federated model. The only difference was that some of the hyperparameters for this model, they've been optimized to improve the model performance. And we used the common evaluation metrics to evaluate the model performance. For example, root mean square error, mean absolute error, and also R squared. Here you can see how these different models performed across scales and also those process points. The blue one are the individual model, the green, the central, and red and orange are federated and federated fine-tuned model. The key findings from this case study was that 750 liter had the most consistent performance across the process points and also other scales. For predicting the feeding time, again, 750 liter was the most consistent. And for end of fermentations, 150 liter and 10,000 liter, they were better for these model developments. Also, you can see that at pilot scales like 750 liter or 150 liter, a federated learning model can be a really good approach if you want to build these predictive models and keep the data privacy. Whereas if you are working at the larger scales, the individual models, they can capture the process variability better. And that's because at larger scales, what happens is usually more complex than the smaller scale. There were also some challenges when we were working with this data. One of the challenges is, for example, when the data are coming from different sites, it's really hard to standardize those data because you need to do a lot of manual work on those data. And also, when you have data from different scales and different processes, the model convergence also is another challenge. And by model convergence, what I mean is that when you are training the model for the federated learning, for example, there is a point that the model is stabilized and then it won't improve after that. So in the federated learning, this can be a challenge a bit. And also, when you are building these predictive models, it's really important to consider the person who is a data scientist and working on building these models. They might not always have the best overview of what happened during the process, so that collaboration between the scientist and the data scientist is really important. And the final word from me is that if we want to make our bioprocess smarter, then we have to start working on these predictive models. And federated learning can be a practical and a scalable method that enables us to keep the data privacy but still benefit from this shared knowledge. And in CPI, we are more than happy to support you if you are interested to work on federated learning, and you don't need to share your raw data. It's, your model can be trained locally, and then it's just the model updates that they get shared. Thank you very much.
Excellent. Thank you very much. I'm going to misuse my position as chair because I want to ask a question to finish us off. And there'll be plenty of time over lunch if any of you have questions as well. So the one thing that I was, I don't know if you can comment on, I know you can't share the data, but how does that kind of process insight that you get, maybe you have a single metric or a single example of where that translates into some sort of process. How much of a process advantage, for want of a vague word, does that give you? So, what benefits do you see typically? I'm trying to be general so you don't have to.
Yeah, so it's mostly if you are scaling up your process and you are moving to the manufacturing, for example, because you need to take these offline samples and go do the lab analysis and then decide when is the best time to start the feeding. So it's labor intensive. Sometimes they have to assign people to stay overnight. So if you can build these predictive models, you can reduce the cost of that and resource management. So the benefits for the manufacturers is in this. But also, this model hasn't been validated yet. So the next step is that you validate this model and then see how it can get integrated with the SCADA system, for example, for the better process control.
Of course. Brilliant. Thank you very much.